everyone, my name is Maleha Bagundang. So today I'm going to discuss our protein experiment. Proteins are made up of many building blocks known as amino acids. Protein are complex molecules and do most of the work in cells. They are important to the structure, function, and regulation of the body. The first test is the coagulation of protein. We have three tests: the heating, NHO3, and ethanol, and give only your observation in each test. Procedure: separate the yolk of an egg to its white, place them into the beakers, then add the same volume of water to each and stir it. Then heat it and observe. The result of the test in heating is after heated, the egg white turned into a bubble, same with the egg yolk. In the test for NHO3, the color changed from colorless to ash gray. In the test for ethanol, it created a smoke inside and has a molecules above. Next is the precipitation by salts of heavy metals. We have two tests, the test for the copper sulfate and lead acetate. Then give only your observation. Procedure: add egg white on the two test tube, then add a saturated aqueous solution on copper sulfate to the first test tube. Then add an aqueous solution on lead acetate to the second test tube. Then observe the colored appeared on each test. The result in copper sulfate is the color became light blue, and for the lead acetate, the color became white. Therefore, color blue and white are indicates the denaturation of the protein. Next is the precipitation by alkaloidal reagents. We have only one test, which is the pictric acid. Procedure: put an 2 ml of albumin solution on the test tube, then add 1 ml of pictric acid solution, then observe. The result is the color became yellow but not include the above part of the solution. Therefore, the denaturation of pictric acid is evidenced by the cloudy materials. The last is the color test for protein. We have 6 tests, which is the Ninhydrin test, Burette test, Santoprotic test, Millon's test, Hopkins Call test, and sulfur test. Since we have 6 tests, we also have a 6 procedures. Procedures in Ninhydrin test. Take 3 ml of aqueous solution of egg albumin in a test tube, then add 4 drops of Ninhydrin solution to it. Then heat the test tube and observe the colored appeared. In Burette test, take 2 ml of protein solution in a test tube, then add 1 ml of 40% NaOH solution, then add 2 drops of 1% CuSO4 solution, then observe the colored appeared. In Santoprotic test, take 2 ml of protein solution in a test tube, then add 1 ml of concentrated HNO3 solution, then observe the colored appeared. In Millon's test, take a small quantity of egg albumin dispersion and gelatin dispersion in 2 test tubes labeled A and B. Add a few drops of Millon's reagent to egg albumin dispersion and gelatin dispersion. Then observe the colored appeared. In Hopkins Call test, take 3 drops of Hopkins Call reagent to a test tube, then shake well. Add 2 drops of concentrated sulfuric acid, then observe. The last is the sulfur test. Add 1 ml of unknown amino acid solution to a test tube using a dropper. Then add a few drops of NaOH solution to the same test tube containing solution. Then put some marble chip into the solution, then heat the solution under the Bunsen burner for 5 to 10 minutes using a test tube holder. After heating, keep the test tube under running tap water to cool the contents. Then add a few drops of 10% net acetate solution into the test tube, then observe. The result of those tests are in ninhydrin test, a color blue appeared, violet in Bury test, orange in xanthoprotic test, brick red in Milon's test, a color red ring in Hopkins Skull test, and a black precipitate in sulfur test.